Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator here at Simulations Plus. I'll be hosting today's webinar, Behind the Scenes, making sure we don't have any technical difficulties. In this webinar, Dr. Brett Pickett, Assistant Professor at BYU and CSO and co-founder of Pythia Bio, and Dr. Jeremy Jones, Principal Scientist of AI-driven drug design at Simulations Plus, will discuss how comprehensive multi-omics analysis can be paired with AI-DD to identify novel disease targets and rapidly develop leads for that target. Sorry, that was a mouthful. Eric Jamois, Senior Director of Business Development, will serve as our moderator today. So a few housekeeping items before we begin. We take our privacy rights seriously by registering for and attending this event or participating in the Q&A. You are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon, and then we'll address all questions in the Q&A after the presentation. Before we begin, I'd like to conduct a quick poll to understand your current familiarity with multi-omics analysis and AI-DD. So I'll launch that. And it is 2 questions. So we'll take a few seconds here. Okay, I see some answers coming in. Give it 3 more seconds.
Good. Thanks, everybody, for participating in the poll.
Yes, thank you. We appreciate it.
And we'll go ahead and end it there. All right, and I'll pass things over to you, Eric.
All right. Thank you. Jasmine, thank you for all of you for joining us. As Jasmine said, I will be the moderator for today. My name is Eric Jamois, and it is my pleasure to introduce our speakers for today. Brett Pickett is the CSO and founder of Pythia Biosciences. Brett completed his PhD at the University of Alabama and his postdoctoral work at UT Southwestern Medical Center, Dallas. He's now part of the faculty at Brigham Young University in Provo, Utah. Professor Pickett focuses on developing and applying big data, AI, and drug repurposing methods to predict therapeutic targets, biomarkers, and diagnostics. And our second speaker will be Jeremy Jones. He's a principal scientist. Jeremy joined us at Simulations Plus in November of last year, focusing on early stage drug discovery and development with a particular emphasis on AIDD. Prior to joining the company, Jeremy was the director of oncology at Terra Therapeutics. And before that, he was a professor at City of Hope, a comprehensive cancer center, focusing on drug development in urologic oncology. So Jeremy received his PhD in microbiology and immunology from Stanford, and also did a postdoc at UCSF. So he actually joined us from really far away in New Zealand. So thank you, and we look forward to some really interesting information for both of you.
Great. Thanks, Eric.
Yeah, thank you.
All right, Brett.
All right, so let me share my screen. Is that coming through okay for everybody?
Yep.
I think it's looking good. Yep.
Great. So thanks everybody for joining us. We're excited to just show you a little bit about how you can simultaneously analyze multiple types of omics data in our software platform, the CDIAM platform. A little bit about the services, but really our goal is to provide this software and integrate next generation pathways, AI and machine learning, and novel workflows that we'll talk about later on during the demo. So there's a lot of capabilities in there, and we'll talk about them from predicting targets for drug repurposing to biomarkers, drug mechanism of action, that sort of thing. So to start, the goal for this first bit is to talk a little bit about Parkinson's disease. I think everybody's comfortable with that, but just as a very high level overview, this is a serious neurological issue. There's a lot of recent findings about the underlying mechanism and which cell types are involved. In fact, we found a paper recently where it showed neuronal cells in this substantia nigra through immunofluorescence actually had some interesting things. So they saw TLR2 as being a component of Lewy bodies, and that contributed to an inflammatory response. So what we wanted to do was kind of walk everybody through a way to look at multiple data types and try to see if TLR2 is possible to be a potential target in Parkinson's disease. All right, so that's the goal. For this first bit, we'll focus on a public single-cell RNA sequencing data set. Just so you're aware, the software we'll talk about can handle single-cell RNA-seq, bulk RNA-seq, Visium spatial RNA-seq, MS proteomics, metabolomics, site-seq, all sorts of stuff. All right, so first off, just a little bit about the software itself. It allows people to really integrate their own data types and analyze them simultaneously, just a few clicks to really try to make sense of all the different types of data, whether it's an expression matrix or differentially expressed genes or just a list of genes that you want to make more sense of. There's a lot of capabilities. We won't go through all of them because I don't want anybody falling asleep. So it does allow differential expression calculation and significant pathways, predicted targets. We'll hit on all those, known drugs, even cell-to-cell signaling and ligand receptor pairs. So a lot of this was built, and we have the capability to add on professional services on top of that if we want to. So the goal today is to go through this Parkinson's disease use case and try to get a better understanding of the underlying disease mechanisms and then how to incorporate and prioritize targets for this really important disease. All right, so like we said before, there's been a few publications, and this isn't even all of them, indicating TLR2 and toll-like receptors in general as having a role in Parkinson's. I don't think this is a big surprise to anybody, but in the past, there's been a lot of work in microglia, and we're going to try to show a new cell type that has that could be interesting to look into more. Okay. So with that, let's move on to what I think is most fun. So this is, let's go to the home page. When you first log into the software, this is what you'll see. So we call them projects. All of the projects that are available, everything with a purple tag is publicly available, so anybody who logs in will be able to see those. In addition, you can make your own projects on the system. And I think it's a nice way to organize things because we don't really want to organize it by data type, like all of the bulk together. We really want to have it disease or indication centric. All right, so I'm going to go into this Parkinson's disease. There's a way to open up the summary. I'm just going to look at the data. So all of the data types that are possible are on this page. I'm going to go into the single cell, and then there's a couple of options. I'm just going to choose the top one because it's annotated. So this is publicly available data. Again, here's the gene expression omnibus identifier, and we've already pulled this into the system. It's pretty straightforward to do. It just takes really a minute or two, whether you're using existing data or uploading your own. Okay, so we do have the capability of looking at ligand receptor pairs from the single cell data. It uses the cell phone DB, the CPDB algorithm. We'll save that for another time. Right now, just to get into analyzing and trying to make sense of this data, we're going to look at it in a pretty universal visualizer. So by default, it's going to come up as a PCA. We're just going to change that to a UMAP. And there's lots of different metadata that we could use to organize this by. By default, it's just by sample ID. So we're going to change this to cell type. So that's always nice. There's a cool legend up at the top. And what I really like about this is there's a way to not just have to manually change things one by one. We can split this panel and do something else. So we could look at, for example, condition and which of these cells, or just change that to a UMAP so they're comparable. So which of the cells were known to come from a patient with Parkinson's disease versus a control? So these were healthy control. I should go into this a little bit more. Sorry about that. These are postmortem samples taken from the prefrontal cortex, and it was Parkinson's versus healthy controls. All right. So if we look at this, the typical players that we would expect are here. We could zoom in and out if we really want to. We'll get into that in just a sec. But if we look at this, it's kind of hard to really make sense of this. But what I really like is we come up here, it'll let us search for the expression of a certain gene. So if we just type in TLR2 and then say, yes, that's the one I want, it's going to change the left viewer in this panel to show up the expression of TLR2 in all these different cells. So let's just give it a second to load all that data. All right, so essentially we want brighter spots. If we look over here, and I'm just going to change this one from condition, we'll get back to that in just a sec, back to cell type. So everything that's over here is going to be the microglia. I don't think that's a big surprise. Most of these already, well, all these patients already have Parkinson's disease, so they are diagnosed. We know the microglia plays a big role in continuation of disease. So that's what the data set is. Down here, there's another cluster, and if we look at that same cluster over here is this little isthmus, so that's more of the astrocytes. There's a subcategory of that. And then there is another one up here that's kind of got a cluster in, if you look over here, these are the OPCs. So for those who may not be comfortable with exactly what that means, we'll talk about that more as things move on. But it is the progenitor cells for neurons. Okay. All right, so just looking at this, I think the microglia is, if we're looking for a relatively novel data type or target for this particular indication, I think microglia is pretty saturated. Astrocytes is not really something I want to deal with for lots of different reasons, but I'm kind of interested in this cluster and what's going on there. So what I'm going to do is I'm going to look at this, because if we look at the microglia, see, it's this one, there's a nice mix of Parkinson's and controls in the same spot, and both of them seem to be expressing TLR2 fairly similarly. This cluster in the astrocytes seems to be some Parkinson's disease, but a fair number of controls in there. But this little bit at the top of this little outlet from the OPCs, that seems to be pretty rich in Parkinson's disease. So that kind of caught my eye. I wanted to look into that a little bit more. So we can either find a marker. In this case, I'm going to look for differential expression, and we're going to run the analysis in real time. So I'm just going to say, maybe I can type coolest analysis ever, because it is. And there is a parameter here to say what percentage of cells do we want to have expressing this gene. I'm just going to set it to 0 to make sure we capture as much as possible at this point. And then we want to set up the control versus the experimental variables. So I'm going to say, all right, for the condition, I want to have the control be the controls and add that in. And over here, I'm going to do something similar. I'm going to say condition, but in this case, PD, and add that in. That's not going to give me exactly what I want. So I'm going to do another set of criteria that will incorporate the cell type. So I'm going to come over here and say, hey, I want these to be OPCs, the progenitor cells, and do something similar over here, and OPCs again. So what this is going to do, and add both of those. Okay, so what this is going to do, as you can imagine, it's going to compare OPCs from control samples versus OPCs in Parkinson's disease, and let us get a better idea of this. Sorry, I'm using Safari, so my settings make me click up there to open up the new window. This is all the analyses I've run. So this is running in real time. It'll just take maybe 5 more seconds, not even that. So now we can click into this and look at what the results are. Okay, so I'm just going to, you can open up the DE, it'll give you a list and a volcano plot. If I run enrichment and just click on that, it'll do the exact same thing. So we'll just skip that step. And if we type in TLR2 here, it is differentially expressed between these two cell types. or excuse me, between the condition in the same cell type, a decent positive log fold change, log 2 fold change, a decent p-value, even FDR is still pretty good. So this makes me curious, what sort of pathways are represented? So by default, it's going to use a hypergeometric. I'm just going to change that to GSEA, and I'm going to enrich against. We support 3 public pathway databases. So we'll use those, and I'm going to add in a filter that says use an FDR value of at least 0.001. So that still leaves me 18,000 genes over all these different entities that are going to be incorporated into the analysis. Our past experience has shown if you can get about that neighborhood is going to give a decent result. So we're going to run that enrichment. This is just going to take a few seconds. If not, I can go back to one that's already finished, but probably 5 or 10 seconds. So up until now, we've looked at Parkinson's disease, the different cell types, the expression of a known, at least a marker potential mechanism that we want to look at, or even a potential target. And this enrichment just finished. Okay, so if we click into this, this gets to one of my favorite parts of the software. I mean, as you can see, we're trying to really integrate the tools with the data and exploration and trying to make scientists' job easier. We're really good at generating data. We're really good at interpreting data. But as far as integrating data, I think that's something that's a pain point that everybody can realistically empathize with. Okay, so here's the basic layout of our viewer. There's a few different components or panels. There's 4 panels. So up here is a visualization of the pathway. Down here is a list of the selected pathway. Bottom left is all of the features that were included in the analysis. Here's a list of pathways. To be clear, this is all the entities that were included in our analysis and everything that's represented in the pathways. What's really neat, this is one of my favorite things, is based on this list of pathways, we've got an algorithm running in the back that lets us find targets that are present in each of these pathways. So I think that's really cool. We call this pathways to targets. And just to show you what it does generate a list of targets. They are prioritized with a default score to begin with. So TNF is first, and then P53 always picks up IL-6. If we look down there, there's a decent list of what you'd expect, right? Including NF-kappa B. Okay, so let's say rather than look at known targets or late stage targets, we wanted to look at early stage targets. Well, we can actually change the scoring criteria for this thing. So we'll come up here to set the scoring template. I've got a couple of these done, but I'll show you what they have. This uses 10 to 15 criteria to prioritize the drug. So I'll kind of give you a peek into our secret sauce. Okay, so you can look at a location in the cell, so the subcellular location, if you were thinking in gene ontology terms, how much weight we want to give to a pathway, how much weight we want to give to protractability. So this is how many targets across all possible modalities. That's kind of what this is trying to represent. Number of approved, this is drugs, so we want to give that a negative score if we're looking for something that's earlier on in the process. We want to penalize things that have bad safety. Again, if we don't want to look at very common targets, we want to find something that's a little bit more novel. We're not going to penalize or reward an antibody-based drug. And then we're going to reward more of the early phase trials rather than the late phase trials. We can even add in what fold change weight we want to include. So just keep this as is, go back to the table. And now there's this new score of what we define it to be. And one other thing I need to do is, at this point, this is showing all the targets that are found across all the pathways. But I want to narrow this down so it's really only representing the list of genes that were found to be differentially expressed in our analysis of the single cell data. So I'm going to click this little radio button, and TLR2 is number 2 based on sorting the original score. But if we sort by our modified score, suddenly TLR2 is the top hit, which I think is really, really cool. So if we go across at all the metrics for this thing, it'll tell you a little bit about what the fold change was, how many known drugs are out there. This is not competitive intelligence. This is pulling from the open targets database. So there's 2 known drugs in open targets, how many pathways it's present in, tractability, everything down the line. So nothing's approved for this. There are 2 antibodies. It's associated with a lot of diseases, and both of those drugs are in phase 2. And then subcellular localization. So what I like about this is each one of these panels feeds the other. If I click on the name, it'll let me see some information or search it in open targets, or look at, this is actually a really huge feature, is look at any other studies that are that have TLR2 as being important. So I'm just gonna, just for now, show the additional information about the genes. So we pull this in from public sources, which is always great. But if I click on the row, see how everything updates? So the number of pathways updates with the pathway that has TLR in it. If I click on the pathway, it's going to pull that pathway from the public sources and let us visualize it that way. So there's a lot of really neat tools that we can zoom in and look at exactly where TLR2 is. Just a lot of neat capabilities. I should mention one thing that we're really proud of is the algorithm for this. We actually just published it, came out last Friday. So this component, we call it pathway to targets. It's kind of a really neat way to quickly prioritize targets based on whatever criteria you want, and you can set your own thresholds as needed. Okay, so with that, I will turn the time over to Jeremy to talk to us a little bit about how we can, well, I'll let him take his I don't want to steal his thunder, but tell us a little bit about how to find a drug against TLR2 that should be useful. So Jeremy, go ahead.
Yeah, Brett, that was great. Thank you. And I will say that the Pythia Bioscience guys have given me a trial license to play around with the Carpe Diem software, and it is really cool. It's been fun to get to learn it and you know, play around with different favorite targets from my past to see where they might be. And we'll get back to that in a little bit. Let me share my screen. Give me 1 second. Sharing my screen. There we go. Yeah. So again, thanks, Brett, for that. That was really cool. You know, we got to the TLR2 as a target. So we've identified a target. Now what? And so I'm going to talk you through a little bit about how we go about identifying targets and developing drugs around them in our sort of what we call our early drug discovery services team here. You can see just some of the steps that we'll do. We'll do a competitive landscape analysis. And then I will say maybe the most arduous and time-consuming part of this is sort of a data gathering and curation phase where we look for all the information out there, pull it together, clean it up, and put it into one data set so that we can use our machine learning algorithms to build some really useful QSAR models with that data. We use those models in our new AI-driven drug design module within our sort of flagship software, ADMET Predictor software. And we can do multi-parametric optimization as we generate new drugs or new candidates. Certainly when we get that list, we do candidate prioritization, including things like looking for analogs by catalog that are rapidly available in like the enamine real space. And we also do a novelty and patentability sort of search as well. And later on in the process, we won't talk about it today, but we can help with hit to lead and lead optimization. So, you know, when Brett and Tristan, who you'll meet in a little bit, came to me and we're talking about this project and looking in Parkinson's disease, we started going through the processes that Brett showed you. And, you know, we were trying to find a target that would be good for drug development. And I just real quick, you know, what a competitive landscape analysis will look like is client to client basis, project to project. But what we're looking to do is identify programs in industry and academia. And, you know, we're looking at different targets. You don't want there to be too much competition, you know, like multiple programs already in clinical trials or multiple chemicals have already filled for on-tox target issues. You conversely, you don't want a target that really there's not enough to go on. That we really need some more basic biology before we can develop drugs around it. So if there's no known ligands or structures, there's no validated functional assays, we may want to wait until the biology is a little bit more mature. And we're looking for that target that's just right for the project. There's emerging interest in the target. There's possibly some molecules in preclinical pipelines. We have some existing starting point. There's some known ligands. There's maybe a structure or two or an assay. And again, as we were going through potential targets in the Parkinson's disease data set, the toll-like receptor 2, the TLR2, really came out to be the one that was just right and met that criteria. As you saw with Brett's demonstration, there's just a couple drugs starting out in the clinic. There's a lot of involvement in different diseases, but it hasn't been overdone yet. So that's something that we do, and one of the ways we arrived at TLR2 for this project. Now, I also want to say that before we get into the data curation, which I said takes up a lot of the time here, you sort of need to know a little bit about the biology. So I borrowed this figure from Invivogen, not a paid promotion or anything. They've just developed what's become the standard assay for measuring activity of toll-like receptor 2 and other toll-like receptors. And so the biology is this, and Brett alluded to a little bit, but toll-like receptor 2 is expressed on the surface of specific cells, usually immune cells. And it forms heterodimers either with TLR1 or 6. It responds to different pathogens, especially cell wall components of different pathogens. Here you can see some of the complicated ligands, either this PAM2 or PAM3 CSK4 that are used to activate toll-like receptor 2. We talked about how it's really involved in the inflammatory response, and that's its main role here. It signals through NF-kappa B. You can see the two components of NF-kappa B here. And when that's activated, it moves to the nucleus and turns on the expression of these inflammatory genes. And so Invivogen has created this great set of cell lines that when TLR2 is activated, you can have a readout of either this SEEP gene, which provides a nice quantifiable colorimetric assay, or a luciferase gene. So a really nice way to measure the activity. And I'm mentioning this now because this gets into what data we want to include and what data we probably shouldn't include in our data set to build our QSAR models. So the next step is really gathering the data, right? So identify the sources. I'm not going to spend too much time on this. These slides will be available if you're really interested in this particular target. But we start with a literature search for small molecule toll-like receptor 2 antagonists. And here you can see some of the major data sources that I drew from to build our data set, with the original being this series of 26 compounds from 2012. And I want to point out the assay here. It's not that Invivogen assay yet, because at that time it hadn't been developed, but it is similar. So you get that PAM3 CSK4 induced, and here they were measuring a nitrous oxide production in macrophage cells. Likewise, this other series of compounds from 2015, they again stimulated the toll-like receptor 2 cells, but this time they measured interleukin-8 mRNA levels by RT-PCR. Some of these more modern data sets from 2018, you have a series here of 13 compounds, and from 2021, a series of 27 compounds, where they did use this Invivogen assay. And so this very standardized assay makes it easy to compare from lab to lab. One other publication we found from a company called Norapor, they actually developed a series of compounds. In the academic publication, they didn't disclose the structure, but the next, another great place to search for data and structures is the patent literature. And in the patent search, we did find that the Norapor patent revealed data on 51 compounds. Other great places to search are the Campbell database, Shore Campbell database. If you're familiar with them, the big advantage here is that you have the structure information with the activity data built in. You can export it and then upload it to your analysis software directly So you don't have to manually draw in the structures one by one. And for instance, the TX series, the fourth series in that previous slide, all that data was present. So it made it really easy to export and import into our software. You'll also occasionally find other molecules that you didn't find from the academic literature where toll-like receptor 2 was used as a counter screen. For instance, we found a couple molecules where they were trying to develop them as toll-like receptor 7 antagonists, and they used TLR2 as a counter screen. So we were able to add to our data set that way. The disadvantage really is that you have to examine the data closely, because sometimes the data in Chembl is not as exactly as they've labeled it. So you always have to trace back to the original paper. And finally, another great source for information is a protein database or similar databases, where you can get some structures that you can use for docking affinity scoring. And if you have a ligand-bound structure, that will allow for 3D ligand shape matching. We have a very cool algorithm in our software that can do that. I'll say for this project, though, unfortunately, there was no toll-like receptor 2 antagonist structures available. I know I'm spending a little bit of time on this, but it really gets to the point that the data that you're using to build your models is really key to this. So a couple quick warnings. Beware the phenotypic assay data. Is it actually a ligand? You know, for instance, I found this paper with TLR2 signaling inhibitor. And this group just stimulated cells with by infecting them with a virus, and then looked for an NF-kappa B reporter, but there's no real definite connection to toll-like receptor 2. So we're not sure it's actually a toll-like receptor 2 ligand. Probably want to leave that data out of your data set when you're building your data set for your QSAR model. Beware the binding displacement assay. This happens a lot in Chembl, where they will list particular compounds with IC50s, but they're not really IC50s. They're just displacement of a known ligand from the protein. Found a big data set again that was listed as IC50, but when you trace back to the original paper, it was actually an agonist, not an antagonist. And so we obviously don't want to mix the agonist and the antagonist data together when we're building our model. Finally, beware allostery. Our compounds binding in the same pocket. So those neuroporant compounds, 51 compounds, great, but they're allosteric. They're binding at a non-competitive site, where all the others are binding at the competitive binding site. And, you know, the requirements for the shape and the characteristics of the ligand will be really different in those two pockets. So you might want to consider keeping them separate and building two separate models. Finally, for the data curation, are the assays comparable? We want as big a data set as we can, so our machine learning algorithms can build the best QSAR models possible. But there is big differences if you're using different assays. You might not be able to compare them. Often, in this case, we got really lucky. So if those assays are similar, if you remember, I specifically pointed out the assay where they measured the nitric oxide production and the IL-8 mRNA levels in the same very similar setup as that Invivogen assay. We got lucky in that the Norapor paper actually tested two of those old compounds in the more modern Invivogen SEEP reporter assay. And so what they found was the C29 compound in this more standard assay had a IC50 around 44 micromolar. And it had been reported in the other assay to be about 20 micromolar. So about on the same scale, maybe a twofold difference. Feel pretty comfortable including those data sets together, maybe with a scaling factor there of around 2. The CU-CPT series, however, had a big discrepancy from assay to assay. It was found to be almost 30 micromolar in this more modern assay and less than a micromolar in the IL-8 mRNA assay. So maybe you're skeptical about adding that data set to all these newer ones. And that can kind of come down to, in fact, I actually built the data sets with and without that CU-CPT 22 series just to see how it would work. And finally, I just want to mention sometimes you'll get data where they don't have IC50s. Instead, this big data set from Kai et al., they actually reported a whole series of compounds, but percent inhibition at 25 micromolar and 50 micromolar. So we can't integrate that with the other data for which we have IC50s. But what we can do is use that data to create a classification model, a yes-no. And this becomes a really powerful approach. So we increase our data size by adding this data. And at first, we create this classification model. And so any new molecules we're testing or developing will go through and first have to pass that yes/no filter. Is it likely to be a toll-like receptor 2 antagonist? If yes, then it'll go on to the regression model we'll build. And I went down a whole rabbit hole thinking about all this data curation. If you're really interested in being bored, or if you need to go to sleep one night, feel free to check out this blog that you see linked to there, going through in detail all of this sort of data curation that we do. So I did end up using all this data to start building some QSAR models. Within our ADMET predictor software, we have a module called ADMET Modeler. If we have time later, I can show you that, or it's something you can contact me later and I'll walk you through. But we use this to build our QSAR models. First, I just want to say, we make sure the data is all clean, no duplicates, no fragments, or non-standard tautomers, things like that. So we're using a really clean data set to build our models. We build the classification model, as I said, and then we use it sort of as the determining descriptor for the regression model. So for the classification model, we had all the same data for the regression model, plus that chi et al. % inhibition data. So 120 total data points, 55 negative, 65 active, nice balance. And then the regression model, we use the IC50 from that very specific SEEP assay. And we created a separate model for those allosteric compounds. Sorry, excuse me. Again, I can show you how we get to this, but here's what the output of our models looks like. You get this really nice false positive, false negative, true positive dichotomy, with the blue being the training set and the red being the separate left out test set. So you can see we had really nice specificity and sensitivity measures for this classification model. Likewise, with the regression model, we had really nice low root mean squared error, high R squared value, a really nice layout between our training points and test points. So a really nice regression model now to use for our QSAR. So oh, I apologize, got something in my throat. In any event, the next step we do is to use these QSAR models in our generative chemistry algorithm, our AI-driven de novo drug design multi-parametric optimization module. So again, this is another module that's within our ADMET predictor. And the way this one works, I'm going to show you a demo. I think it's easier to sort of do both, see this screen and then see the demo. But the way this works is we take some seed molecules and then we initialize some random chemical transformations, add a methyl group here, change a pyridine to a pyrimidine, things like that. We make all these analogs, and then we evaluate all the specific properties that we want. We have a whole list of different properties, but some of the normal ones we use are ADMET risk. We have this great ADMET risk score, which brings in 22 different ADMET factors into one risk score, a synthetic difficulty score, our QSAR models here for activity. We have HTPK, high throughput PK predictions, so we can actually predict things like oral availability right up front. And we have this ability to do 3D shade match scoring. If you have any programs like a docking score that you want to bring in as well, we can integrate that score with this multi-parametric optimization. So we generated these analogs, we evaluate all these properties, and then we prune them using Pareto optimal layers. So basically, the best in any one of these dimensions, in any one of these properties, get saved and then used to generate more analogs. We go through this process. A typical run will be, 40 or 50 generations. And it'll spit out a list of molecules at the end that are optimized in these multiple properties. So, oops, sorry. I'm going to show you just one potential AID campaign. You know, you can imagine multiple different things you want to do, but looking specifically maybe at that TX series. We use our ADMET predictor tools to understand the QSAR. We can create classes and perform R table analyses to see what different R groups affect the activity. A very powerful tool we have is matched molecular pair analysis that you can see an example of here that will show you what small structural changes have an effect on your property of interest. In this case, the PIC50 for inhibiting TLR2. I'm battling through it. Don't worry, folks. So you can see two examples here where pyridine has much higher activity than the pyrimidine. So that's something that we want to keep in mind as we're designing new molecules. We saw other things using this, like a Paris substitution on the pyridine reduces activity. But one thing we noticed was that there was plenty of room for exploration on this central ring. So for our first strategy, we decided to explore the SAR on that central ring. For our optimization parameters, we'll use our QSAR models, the ADMET risk score, percent bioavailability, and synthetic difficulty. Because these are Parkinson's disease drugs, or we're hoping them to be Parkinson's disease, we also added a filter to make sure that we have a very good classification model for predicting whether a compound will pass through the blood-brain barrier. So we set that filter to make sure these compounds had a high blood-brain barrier penetration. For a seed molecule, we'll use this TX37 molecule, which was the most potent in that series. And we'll create a query filter that will really focus the evolution of the molecule on this central ring. So I have a full webinar about this AIDD module. If you're interested, look here. But for now, I'm going to move over to this. This is what the compound data set looks like in ADMET Predictor. You can see we've entered the PIC50 information, whether it's active or not. And again, if you're interested in all this cool stuff that we predict about every compound, please check out the Simulations Plus website to find out some more. But I specifically want to show you one run of AIDD here while I have a little bit more time. So that seed molecule we were interested in is this. That's TX37. You can see it's a submicromolar compound, active. And so all I do here is click on this AIDD button. And I've pre-populated some of this, but you can see all the different parameters that we have that we could optimize at this simultaneously. As I said, the ones we were most interested in here, we want to minimize the synthetic difficulty. We want to minimize the ADMET risk score. We want to maximize the fraction bioavailable. And we want to maximize the PIC50. In the next slide, this is where we can define a scaffold query. So as I said, we want to focus the evolution on the central ring. Let me just show you how we got to this. So these weird symbols here, basically for any of the atoms, we can bring it up and we can dictate what symbol it is, what charge, what isotope, what hydrogen substance, how many rings we want. And this is what I did, set the substitution count as drawn. So that means it's going to stay the way it is for all these molecules. And so only these ones will be allowed to be changed. We have a drug-like filter that makes sure if we make any sort of ridiculous molecules, they'll be filtered out. Last slide here before we hit go. This is our transforms rule. So you can actually see all the different transformations we allow. You can inactivate if you want to or not. And then you can set the number of optimization generations, the candidates per generation, and using multi-threading, which means it'll use as many processors as we can to make this process faster. As I said, Normally we would do like 40 or 50 generations, but just for time, I'm going to set it of just a few molecules in 10 generations, and you'll see really how fast this is. Hopefully it works, right? So you can see we're already sort of on generation 6 and 7. Not only is it processing all these, it's changing them into 3D conformers as well, because some of the models we have are built on 3D descriptors. Now, if we're lucky, the new window will pop up. If not, I have one. Oh, here it is. Perfect. In the live webinar, I'm always nervous as to whether or not it's going to work. But, oh, it worked. It worked fantastic, like it always does. So what you can see here is this list of new molecules we've generated. Here was the seed we used, and you can see the different properties. So the percent bioavailable, the PIC50, predicted the ADMET risk. So our seed molecule had an ADMET risk of 4.8. If we wanted to sort, we can find some molecules that still have a nice high PIC50, but a lot lower ADMET risk, still very bioavailable. And then you can sort by any properties here. You can filter to make sure you want them all above 50% oral availability. The IC50s, let's make sure they're all at least 10 micromolar or better. ADMET risk, want to make sure it's low. And synthetic difficulty. And so you can filter really quickly and easily to help with compound selection this way. There's so much more this can do, but for now, that will be the quick demo of AIDD. Let me go back over here. What happens after this? Well, you know, we would dive into the design, make, test, analyze cycle. We can do candidate prioritization. So we have a new tool in our newest version of ADMET Predictor coming out, a multi-criteria decision analysis tool, where you can weight, you know, pick as many properties you want, add a specific weight to it, and it will rank the compounds that you've designed based on that criteria. So, you know, if you wanted to 75% of the weight on the PIC50 and 10% on the ADMET risk score, it'll rank them that way for you. We have the ability to do rapid, you know, an analog by catalog approach. So see if any of the molecules we've designed or very similar molecules are available in commercially available synthesis on demand libraries like the Enamine Real or the Wuxi Galaxy. We have a novelty assessment that will sort of de-risk and make sure you're working in a novel chemical space. We're actually working on building that in as an optimization parameter right now. And then, of course, after we test, we would use ADMET Predictor and AIDD for further rounds of design and optimization. So a couple points I want to make here at the end is that, you know, most drugs don't fail in the clinic because of affinity or activity issues. Most of them end up failing because of ADMET or PK issues. So to us, to me, I think to us and hopefully to many other researchers, it's really important to incorporate those ADMET and PK considerations in the earliest stages of drug development. And you can see that as we're designing the molecules in AIDD, we have an eye toward that, making sure the ADMET risk is low, the oral availability is predicted to be high. So you don't have to worry about all that optimization later, where you might risk hurting the activity. That being said, those predictions are only as good as the data and the models on which they're built. So there's, you know, publicly available data sets to predict some of these properties, but there's nobody, you see the care and time we take, just even with this QSAR model, making sure our data is curated. We do that for every one of our models. To really make sure that we have the best predictions and that it really does influence the how good the drug will be as it moves forward in the process. So a short little intro about what we do and how we got to the TLR2 antagonists. It's a project that I hope to continue forward. But I do want to say, you know, Brad, I'm going to toss it back to you here in a second. Hope you're still awake. Hopefully I didn't put you to sleep. But, you know, in that other webinar I talked about, if you go back and look at it, I was designing PPAR gamma antagonists for bladder cancer. Just real quick, one slide. You can go see more about it. But just to point out that 20 to 30% of advanced bladder cancers have PPAR gamma pathway alterations. PPAR gamma is a nuclear receptor transcription factor. Again, always important to know a little bit of the biology there, but most important here is that a couple years ago, a bunch of papers came out showing that this is a really important target in bladder cancer. And so people have just recently started to develop some PPAR gamma antagonists for bladder cancer. The first one started a phase 1 trial just a couple months ago. I used the same process that I just went through for toll-like receptor 2 to design novel, potent PPAR gamma antagonists. The whole data curation process, including some data from my old lab, building the QSAR models, doing AIDD to generate some new molecules. And so obviously we're interested in developing this for bladder cancer, but we wanted to know if there's an opportunity to expand to other indications. Is PPAR gamma important anywhere else? And this is part of this partnership with Pythia Biosciences. They have the ability to help with this. So Brett, I'm going to turn it back over to you, if you don't mind. I got about 5 minutes.
Great, thanks. All right, thanks for that. That was amazing. I'm always a big fan of AI, especially when it works and does what you want to do. It's really impressive stuff. So hopefully y'all can see the right screen. Wanted to follow up on that. And so yeah, PPAR gamma is important in bladder cancer. We were interested in, hey, are there any other indications where PPAR gamma could be relevant, especially antagonists for this thing? So again, we brought in HCC data set, hepatocellular carcinoma, and we wanted to play with that in the Carpe visualization platform. So again, this was based on this paper. So went out, grabbed the DEGs, took about 30 seconds to import it into the system. And once it's there, any project and all of the data sets in a project, you can really just use the search bar and see if PPAR gamma or any gene is present in that data set, which is could be bulk or single cell or proteomics. Again, anything, it's going to be indexed and searchable. So we did that. Surprise, surprise, PPAR gamma was relevant in HCC. So we wanted to look at the differential expression of that thing in the bulk RNA-seq. And kind of a bummer, it was not as good as we had hoped, especially based on the paper. So if we looked at a log fold change of this, 0.17, and then a really bad FDR adjusted p-value, most people would have said, okay, no, I'm out, right? But with this system, we imported another data set. It was a single cell data set. And so the question was, is this real? Or is this because of all the heterogeneity in the cells that make up the sample that was actually included in the RNA, in the bulk RNA sequencing? So we brought in this single cell data set and just visualized it with half violin plots. We're just going to focus on the tumor and the normal cells. They had a lot of other cell types and samples, and then we divided this out by cell type, right? So we were looking at hepatocytes, since those are the cancerous ones, and wanted to see if, well, just looking at this graphic, you can see there's a really fat tail on this thing. So PPAR gamma is expressed a lot more than it is in the normal cell type, in normal hepatocytes. Okay, well, that's something. But then we ran differential expression on tumor versus normal in this data set. And if you look at this, the log fold change is 2.5, which is, well, so it's upregulated really high. That's extremely high for a transcription factor. Usually they don't change a whole lot. And then the FDR, 10 to the minus 100, I don't know, I think that's acceptable. So suddenly this starts to become interesting. All right, we've got a single cell data set. We know which cell type is really contributing to the signal. It's kind of getting drowned out in all of the bulk RNA-seq. But we also wanted to play with another data type. So we brought in some cosmics data from NanoString. All right, yes, this is supported too. It's clearly takes up a fair amount of storage space to get all this data in there, but it's really useful to help with trying to justify a go or no go decision. So if you look at this, the tumor tissue or tumor cell types are kind of the weird pinkish purple. I'm never good at describing colors. And there's a different tumor cell that's represented in orange. Then you've got all the other cell types that are also represented here. Okay, so just looking at this, graphic isn't nearly as informative as looking at a violin plot or a half violin plot. So in this one, we've got all the different cell types, and we're just looking at expression of PPAR gamma in all these cell types. It's not super great. And then you look at these, this is actually the gray and the brown are both tumor types, and they're sky high. So again, this is upregulated in the cells of interest, in the tumor of interest, and it justifies really having exploring more of a whether an antagonist could really help with this particular cancer type. So hopefully this was useful in showing you there is some strong evidence that suggests PPAR gamma could be relevant not only in bladder cancer, but also in HCC and likely in some other oncology indications. So with that, I'll conclude this part of the seminar. Thank you all for your time and turn it back over to Jasmine for the Q&A. So thanks, everybody. Appreciate it.
Thank you. So we do have one more poll before we go to the Q&A. I'll launch that now. Just 2 questions, and I'll give everybody a couple seconds there. All right, I'll go ahead and end it there and turn things over to Eric.
All right. Well, thank you, Jasmine. And thanks to all of you for following us through those presentations. Thanks to Jeremy and Brett for some really interesting material and pretty fascinating how you can go from target to actual compounds. Either you hear one or the other, but it's rare to hear both in the same presentation. So that was really cool. There's maybe one question either for Tristan is joining us, and he is Tristan Gill is the CEO and founder of Pythia Biosciences, so he's joining the Q&A expert panel. So thank you, Tristan, for joining us. A quick question maybe on the target selection. So as we know, there's often some redundancy in the pathways that when you take your target and you try to shut this one door, you can have other doors that open to make up for it. So I didn't quite catch in the target selection process. Is there a way to see the level of redundancy, if there's any redundancy in that pathway, that if you affect TLR2, you're not going to have some other process make up for it? I guess that's the question.
Yeah.
Who wants to take that one?
I'll give it a go, and anybody else can fill in the holes.
Sure.
I think that's an excellent question. It's really, as you all know, there's a lot of redundancy, especially in TLRs. We've done some work in TLR 7 and 8 in the past, and there's a lot of crosstalk between those two. So I'd say it's really context dependent. The only way to really test that would be to do an experiment or to find data from somebody else who did the experiment. You can look to see how many pathways TLR2 is involved in. You can see if there's some redundancy that way, but at the end of the day, we haven't annotated every gene that's participating in every pathway in every cell type. So it's really it's got to be done experimentally.
Yeah.
So network strategy will help with that as well.
Yeah.
But in the end, you got to get this into the wet lab if you're going to progress something. And, I think we set you up pretty well for that. And if it's going into model systems, we can help with that and help you track what you're seeing out of your model systems as well.
Yeah. So I think the goal of this is integrate a lot of data together and help you to make a more informed decision or construct a more informed hypothesis that could then be tested with more computational work or more wet lab work or really both.
So there was a question that came in a panel on the ability to analyze different omics data sets simultaneously on the platform, proteomics, metabolomics, etc. So I don't know how you can, mix these different analysis together. Care to comment on
absolutely.
that?
And that's really the whole, the whole premise we started with Pythia is to build this from the ground up. Everyone's been talking about breaking down data barriers for 25 years. No one's done a great job at it. So we built it from the ground up. We actually have a really nice white paper on colorectal cancer, a really nice paper on Parkinson's disease. Both of them talk about some of our integration strategies. So lots of different ways to integrate this data together. You can integrate it at the pathway level. We have pipelines that help you do that. And you can also create unique multi-omic signatures, which is a really interesting way to find the full picture that's going on in the biology.
Thank you. I think we're coming right on the hour. So there are, maybe it's the proper time to wrap up with some conclusion, Jasmine, and we'll, whatever we did not answer interactively, we can get back to those specific attendees, and address their question one-on-one.
Yeah, I appreciate everybody hanging in there and hanging with me while I was struggling a little bit there with coughing. I do appreciate it.
I had a glass of water next to me, but that's really far for you.
Yeah, it's across an ocean or two.
Indeed.
All right. Thank you, everyone. For any questions we weren't able to get to, our BD team's going to reach out. We invite you to visit our website to learn more about our AIDD capabilities. I put a couple links in the chat, and this webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you, everyone.
All right. Well, thank you to all of you for participating.
Thank you.
Thanks, everyone. Great job, Brett. Great job, Jeremy.